Hi, welcome to the Bioinformatics Chat. Today my guests are Keith Slotkin and Kaushik Panda. Keith and Kaushik are studying mobile DNA, and Keith recently published a paper titled The Case for Not Masking Away Repetitive DNA. And so we'll be talking about repetitive DNA and transposable elements and try to get a little bit of education on those very important concepts. Gentlemen, welcome to the podcast.
Thank you very much for having us.
Thank you, Roman, for having me as well.
Okay, let's start with your backgrounds. I'm curious how you got to study transposable elements. Kaushik, let's start with you.
I'm currently a postdoctoral associate working with Keith. I joined Keith's lab as a graduate student back in 2011. And back then, this concept of transposable elements was very interesting to me because traditionally, I was always taught with this very specific positions of genes, and the idea that there can be something in the genome that can move around and cause multiple copies was very interesting to me. And then I had some background in computer before I joined Keith's lab. So I was quickly absorbed as a bioinformatics person and transitioned from a wet lab to bioinformatics and start working and understanding how are these repetitive regions silenced and prevented from affecting a genome.
That's awesome. Keith, what about you? What was your path like?
Well, my path was originally, when I was a graduate student at UC Berkeley, I was already into plant biology, and I joined a lab to do a rotation that was studying epigenetics, and the mentor there, who I ended up following to do my graduate work, a guy named Damon Lish, he just started me on this project, and he started explaining transposable elements and repetitive DNA to me, and I had never heard of it. And I thought they were really cool, and the way he explained it was kind of like the bad boys of biology, these kind of things that people would ignore, they wouldn't talk about, but they were responsible for a lot of new gene creation or changing rates of mutation, which can really change the rate of evolution. And he really sold them to me, and I've been studying them ever since. I studied them as an undergraduate, a graduate student, postdoc, and now obviously in my own lab. But being both a plant biologist and studying transposable elements, I'm doubly studying something that a lot of people ignore. So there's
That's
probably
very true,
yeah. So there's probably something there that I like in my personality, that I like studying the things that have slipped under everyone else's radar.
Let's start with the basics. What is a repeat and what is a transposable element? What's the relationship between those two concepts?
Well, a repeat is very broad. It's just something that is going to be in a particular genome multiple times. There can be genes that are repeat, where the gene has been duplicated a number of times, and then there are things like tandem repeats, microsatellites, and then as a subcategory of repeats are going to be the transposable elements, which are broadly defined as fragments of DNA that can have the potential ability to mobilize or duplicate themselves from one region of the genome to another and to insert into another part of the genome. And there's lots of different flavors or types of these transposable elements that all use different mechanisms. But we use transposable elements as a blanket term for all of these elements with that particular ability.
Right. So what are some of these mechanisms? How does this transposable elements manage to copy themselves?
There are some mechanisms that are RNA-based. So the element will make itself into an RNA copy and then make a what's called a copy DNA or cDNA copy of itself and integrate into the genome that way. Those are called retrotransposons because of its retro to the current the central dogma of DNA to RNA. So this is going back from RNA to DNA. There are DNA transposons, which create proteins that pick up the whole element as a fragment of DNA and transpose it into the genome. And there are a number of different types of mechanisms of those, including something called a helitron that does it a little differently. But basically, they can be classified on how they move, and then how they move is reflected in their structure of DNA sequence.
So what triggers this process? Because if these transposable elements would be copying all the time at the same rate as, for example, transcription or replication happens, then our genomes would just grow uncontrollably and become very huge. So clearly this is not, and also the genes would get disrupted when these transposable elements get themselves inserted inside the genes. So this is not happening too much. What is stopping them? What are the constraints?
There are specific mechanisms that the host or the genome actually employs to try and stop the transposable elements from doing that. By default, they would like to make as many copies of themselves as possible. But there is quite a bit of investment on a cell's part to try and identify which are the transposable elements which are making multiple copies. And there are multiple levels of silencing. They can be stopped at the transcription level, prevented from making an RNA transcript to begin with, or their transcript can be degraded later. There are DNA methylation mechanisms which specifically tell the cell that these are the regions that needs to be shut down pretty strongly so that they don't make copies of themselves. But there can be multiple environmental triggers, or like sometimes there are also examples of specific mutations in these silencing mechanisms that might actually allow the transposable element from being expressed.
Right, so there's like evolutionary pressure against these transposable elements being active all the time. So the cell is sort of aware of their presence and tries to silence them.
Yeah, the analogy that's been made is very similar to a virus versus its host. It's an evolutionary arms race where the host is trying to repress transposable element activity. The transposable elements are trying to do what they do, is to transpose, and there's an arms race in identifying new mechanisms and then counter defenses to those new mechanisms to in this evolutionary battle for activity versus silencing. That analogy ignores some of the real benefits that transposable elements can have to a genome, but nonetheless is a useful framework that we often use in the laboratory.
What are those benefits?
Well, a transposable element, the process of transposition itself is inherently sloppy, and transposable elements often pick up or co-opt fragments of genes and can rearrange those genes or mix several parts of different genes together. And there's cases in which transposable elements have created a novel gene with different domains that never existed before because of their gene scrambling ability. In addition, famously discussed by McClintock, who discovered transposable elements, there's this idea that transposable elements, which can rapidly affect the rate of selection in evolution because of their changing the rate of mutation, they can activate under times of stress and potentially help an animal or plant or fungi overcome that stress by creating mutations and hitting on that 1 in a million mutation that may help overcome that particular challenge.
So you made this analogy between transposable elements and viruses usually have to encode the machinery needed to, for example, embed themselves into the DNA. But these transposable elements, are they similar in that they try to encode this transposase, or do they rely on the cell to perform this function?
They encode the proteins necessary for their transposition. So I want to make a distinction that we differentiate transposable elements into two categories. What's called autonomous or auto, meaning that it can replicate itself. It produces it produces this proteins necessary to replicate itself. And then non-autonomous, which is if those proteins were made by an autonomous element, they could act on the non-autonomous element to transpose it. So there's a master-slave analogy here where you have the master elements that can mobilize these other ones as well. And in the genome, in any genome, the non-autonomous elements, I should say any eukaryotic genome, the non-autonomous elements there's many more of them. They outweigh probably 100 to 1, if not more, the actual autonomous elements that can transpose themselves, because transposable element fragments are essentially everywhere. But the proteins that they often encode will just do the basic function of mobilizing the DNA. They often don't they don't carry the proteins to finish the process. They rely on the cell's repair mechanisms of its DNA, which are host encoded, to kind of clean up the transposition process to finish, whether it's to ligate the DNA or finish the base insertion to make the perfect insertion site. So they require that, maybe let's say the cutout mechanism and to insertion mechanism, but not the finishing, polishing of the DNA. They rely on the mechanisms that are already around for that.
So these transposable elements then, they actually encode proteins, especially the autonomous ones. Do they, like some viruses, can they encode multiple proteins in the same sort of transcription unit? Or is it the case that multiple transposable elements need to be transcribed in order to activate this machinery?
No, they can actually encode multiple proteins, and sometimes the RNA transcript can be spliced, and that can give rise to a different transcript, give rise to a different proteins. And so they can encode 4 to 5 different proteins that they require specifically for their processes. Not all of them have to be functional, but that kind of determines whether or not that transposable element actually moves.
And do we know anything about how these things arose? Are they just very ancient viruses that embedded themselves into our DNA, or do they arise inside the DNA itself?
Well, this is a topic of great debate in the field. So we know there are cases in which transposable elements have come from disabled viruses. So a virus can replicate itself and then leave the cell, typically packaging itself in a protein coat and then infect another individual. So transposable elements are often generated from these viruses when they lose this ability to leave the cell, but they can still replicate themselves and insert in new places in the DNA. So we have lots of examples where a virus without an envelope protein to leave the cell is now a transposable element. On the other hand, we know cases in which a transposable element has picked up a new function and become a virus. So there's a back and forth there that it's very difficult to go back to the very beginning, and is something that people discuss vigorously at the transposable element type meetings.
That's very cool. Can you tell us more about that transposable element that became a virus?
Transposable element that became a virus. There are cases in which people have identified a transposable element. These are typically retrotransposons that are already similar in structure to a retrovirus like HIV, for example. And the retrovirus has picked up a new coding region. Transposable elements love to pick up pieces of DNA and rearrange pieces of DNA, and those can be genes. And if they pick up a region of DNA that has a selective advantage for that transposable element, it will obviously be selected for and used. So that process is called exaptation or co-option, where they're using something for a purpose in which it was not originally evolved for in the cell. And so you can have cases where now the transposable element can pick up a new function. This is a new weapon in that arms race that I was talking about, and potentially leave the cell and infect another cell.
My understanding is that these transposable elements, when they manage to activate themselves, they get transcribed by RNA polymerase 3, as opposed to RNA polymerase 2 that transcribes most of the genes. Can you talk about the significance of this? What does this mean for this whole process?
Well, autonomous transposable elements, ones that make proteins, those protein coding transcripts need to be produced by RNA polymerase 2. RNA polymerase 3 replicates sign elements, which are the non-autonomous family counterparts to line elements. Line elements are the retrotransposons that are highly abundant and can be active in human, mouse, and chimpanzee primates, and mammals. So these line elements really dominate our genomes, our human genomes, and they give rise to signs, these small non-autonomous transposable elements, which there are just tons of in the human genome. And these signs are duplicated using RNA polymerase III. So this is not a general transposable element mechanism in which Pol3 or RNA polymerase III replicates transposable elements. This is very specific to a transposable element type that is highly abundant in the human genome.
Got it. Yeah. But how does whether a gene, I guess, or a gene-like element, whether it's transcribed using RNA polymerase 2 or 3, how does that affect its fate? You said that these lines, they have to be transcribed by RNA polymerase 2. Why is that the case?
Well, the lines are protein coding. So to make that polyadenylated mRNA transcript that's going to be destined for cytoplasmic export and translation, those are Pol2 transcripts. And so those Pol2 transcripts are actually absolutely required for the protein that's necessary to move the non-autonomous element. The sign elements being regulated by Pol3 maybe, this is speculation, but it may be advantageous because Pol3, as far as we understand, those types of promoters aren't under the same level of spatial and temporal regulation as a Pol2 promoter. I don't know as much about Pol3 promoters, and I'm sure there's people out there that would argue with this, but the Pol3 promoters that we've used in the laboratory seem to be generally constitutively expressed at a pretty high level. So they don't have the dynamic regulation that a Pol2 promoter often has. So Pol3 expression is just, pretty high and pretty much everywhere. And that may be a general mechanism of evasion of regulation that's based on Pol2 transcriptional control.
Ah, that makes sense. But does that then mean that these signs, they are being expressed all the time? Do they just do nothing there, or do they do anything?
Well, there's so darn many of signs that it's hard to put them it's hard to say one thing for signs because there are in the tens of thousands of copies. And so people have broken them down into subfamilies and studied those individual subfamilies of signs, like ALU is a really, ALU is a very commonly discussed sign element. So I know signs absolutely can be expressed even when they're not transposing. They can absolutely affect the transcription of neighboring genes or genes in which they sit in the UTRs or the introns. They may not. It's very difficult to really understand their specific function, but we now know that a lot of these signs, when they transpose and locate near genes, they can evolve into things like regulatory domains or protein binding sites and affect the transcription of that promoter and may even become a promoter regulatory element.
So we've thrown around these abbreviations like signs and lines. Let's dig into them a bit deeper and let's take them one by one. So let's start with the lines, right? What does that acronym stand for? And what can you tell us about it?
Lines is actually an interesting story. I mean, I've never read this, but this is what I've heard, is that, line has a specific definition, long interspersed nuclear element. And, but the story that goes along with this is that when people were studying the human genome and doing Southern blots, what you would do is take total human genomic DNA, digest it with a restriction enzyme, and run that on a long gel electrophoresis. And if you just look at that gel, it's a general smear because you have so much DNA of all these different sizes, but you then have very specific lines that appear on the gel. And what are those lines? Because they're pieces of DNA upon this restriction digest that are piling up. These are repetitive DNA. These are the transposable element. And lines make up, I think the number is 23% of the total human genome are long interspersed nuclear elements. So huge percentage of much more than the percentage of the human genome that's made up of all genes, for example. So that's where lines came from. And then once they had lines, which are the long ones, then they had signs, which were the short ones. So short interspersed nuclear elements. And so there's a lot of naming transposable elements, I should say, is absolutely the wild, wild west. They have some really funny examples. There's a very well-studied retrotransposon studied by a lab that initiated in Spain that was called Gypsy. And then there was a locus that they identified that controlled gypsy movement. And so they called that flamenco because flamenco made Dipsy jump or Dipsy dance. And so there are lots of cases. It's basically if you discover a transposable element, you get to name it. And so there's all sorts of interesting or funny examples of hobos or mutators or, the original McClintock was ACDS, which was dissociation, and then the activator of that dissociation because the chromosome kept breaking at that site. It can be maddening to people when they start out because we now have systems in which we try to code them in a hierarchical, logical way of what type of superfamily, what type of family, what type of element this is. But if you're just looking at the name and you find a transposable element that's called jockey, for example, that tells you nothing about the organism or what kind of transposable element. It was up to the person who found it to name it.
Do you classify these elements based on their history? Like you said, you can classify them into families and subfamilies. So how does that work?
So we don't classify them based on their history as much as their type of element. So typically there's a coding system that people have created, a 3-letter coding system, which would start with like a D or an R. So is it a DNA transposon or a retrotransposon? And that's the very, very first distinction. And in almost all cases, you can just look at the sequence of a transposable element and say, is it encoding a transposase-like protein? Or look at its ends. Are they direct repeats or inverted repeats at the ends, and see that an element is a DNA transposon versus a retrotransposon. Okay, so that's the first subcategorization that can be done. And then from there, you can say, okay, well, is it, there's 2 major classes of retrotransposons. There's what are called the line elements, and then there are the LTR retrotransposons, and that stands for long terminal repeat. And these have long terminal repeats at their ends, and those are real easy to spot by looking at their sequence. And these lines and LTR retrotransposons have different mechanisms of transposition, although they both use RNA. And so that's a real easy distinction that can be made there. And that's the second letter typically is either like an L for line or a G or C for gypsy or copia, which are the 2 major families of LTR retrotransposons. So it's very much hierarchical, and you can get down into the nitty gritty because after these 3-letter codes, you can add a number. So people have created these systems for hierarchically classifying transposable elements, but that can get difficult because you might have DTM43, DNA that encodes a transposase that's a mutator type, and it's number 43. But we also know that as MUDR, or something that we've given a common name to, and that's been around in the literature for a long time. So now all of a sudden the transposon has two names, one that's, interesting, and the other one that's a coded name.
So you mentioned before the association between lines and autonomous transposons. What is the exact relationship? Are all autonomous transposons lines, and are all lines autonomous transposons?
No, autonomous transposons can be DNA transposons as well if they encode all the proteins that they require to jump themselves. So that's the definition of autonomous. Lines is just one kind, specific kind of transposable elements, and most of them are autonomous.
Yeah, in lines, there are specific subfamilies of lines that we know now are highly active, and there's lots of other. I mean, I can't stress the fact as much that 99% of the transposable elements that you probably find in any eukaryotic genome are likely going to be mutated or derivatives or fragmented elements, and you get the long, full-length autonomous element somewhat rarely. Now, in some cases where particular genomes are going under a burst of transposable element activity, and we have examples of that, you'll find more active autonomous elements, but transposition is so messy that you have all of these fragments in the genome, and because most genomes don't have very tight constraints on their genome size, that they're allowed just to essentially be part of the genome and be carried forward, even though it's a broke down, old junkyard piece of transposable element.
You brought up this notion of being active, and active transposons is one that has a chance to transpose somewhere else or jump somewhere else. What differentiates active from inactive ones? Is that just they get damaged in the process and become inactive, but all the intact ones are all active?
No. So even the intact ones can be silent. So the thing is, the autonomous transposable elements that have the capability to jump by themselves, but that does not necessarily mean that the cell allows them to jump. As I was talking about those silencing mechanisms earlier, they can be in place specifically restricting these autonomous transposable elements from jumping. And it's the same thing with the non-autonomous transposable elements. I mean, if the autonomous transposable elements does not express themselves, make those proteins required for transposition, the non-autonomous transposable elements cannot jump. And so actually one of the recent publications that we've had a couple of years back, we hypothesized that there are specific mechanisms that separately act on the autonomous and the non-autonomous ones, because somewhere the cell might recognize that as long as it acts on these autonomous transposable elements, it has a pretty good chance of shutting down the transposable elements from expressing.
Oh, that's pretty cool. But once this autonomous transposon escapes these controls by the cell and manages to be transcribed, is that the case that it can lead not just to one new instance, one new copy, but as you, I think, said before, like a burst, like there could be 100 more copies suddenly appearing in the genome?
Yes, the evolutionary history. So when we look at a genome today, even though it might not have active transposable elements, we can see evidence of punctuated activity of transposable elements, which we call transposable element bursts. And people have devised all sorts of very interesting tools to date when that happened. How long ago did this burst occur? There's two types of ways that can occur. One, which is more mysterious, is that suddenly an existing element within a genome can activate, let's say, get a new weapon in that evolutionary arms race and now defeat the host silencing and now go under a burst of activity. And this can massively expand a genome in size. And it can produce many copies of itself, all of which themselves can be active. Or you can have a transposable element, and there's examples of this, that's horizontally transferred. So it was never in that genome. So the genome doesn't have a defense against it, but it was part of DNA moving from one organism to another, and often not highly related organisms. The example I'll bring up is the little brown bat genome, which is a mammal, carries a transposable element that has massively amplified in its genome, but looking at other bats or other mammals, nobody could understand where that came from. And we now think that comes from the insect kingdom, probably through a parasitic mite or some sort of flea or tick that was in a close proximity to the bat.
So Keith, and you said you also studied plants, and plants are famous for having a large genome. So does that have anything to do with transposons?
Yeah, absolutely. Some plants have very large genomes, often are crops, and the plants we're most interested in agriculturally have large genomes. And that's really, there's two reasons how a large genome can happen, and they're both at play here. One is transposable element activity. So in the case of the maize genome, for example, which has a large genome, there's been a lot of transposable element activity, particularly from LTR retrotransposons that have massively expanded the size of the genome. And the other mechanism is if you duplicate genomes and you have more than one set of chromosomes in that genome. So that's the case, for example, with the wheat genome. And the wheat genome is a hexaploid. And in agricultural plants, there's been this not purposeful selection on large genomes, because when we have more genome content, we know that the cells are larger, and that then translates to a larger product yield of the plant. So in the case of strawberry, the wild strawberry is a diploid organism, but people over the years, sometimes knowingly and sometimes unknowingly, were selecting for larger fruit, larger yield, and for wheat, the same is true. And so they selected for duplications of chromosomes and of whole genomes, which produced larger fruit. So the current supermarket strawberry that you would find is an octoploid. It's got 4 sets of 2 chromosomes. So in plant genomes, particularly agricultural plants, there's been almost an accidental selection for large genomes. And the way you get a large genome is you either duplicate whole chromosomes or fractions of chromosomes, or you cross-hybridize with another plant and get multiple sets of chromosomes, or you massively activate transposable elements. And often these are non-mutually exclusive. These all go on at the same time.
Oh, that's very interesting. But it's not the case, right, that simply having a large genome somehow causes bigger cells. It's probably the other way around, that bigger cells allow a presence of a bigger genome.
Yeah, that could absolutely. I mean, we know that there's a distinct correlation between, if we start doubling genomes, so we have a tool in which we can let the nucleus, I should say, go through cell division, but use a drug that doesn't let cytokinesis happen. So essentially you can double pairs of chromosomes. And we know that there's a correlation between how many chromosomes are in that nucleus and the final cell size. It might be cause or effect, but nonetheless, it's something that has been unintentionally selected for just looking for larger yield for, thousands of years. Wheat is the perfect example. Wheat is a hexaploid compared to its normal diploid ancestors.
So we talked about lines. Let's now move to their shorter counterparts, the signs. First of all, where's the threshold that delineates lines from signs?
Oh, they're very different. The only thing that really ties them together is that lines are the autonomous transposable element responsible for mobilizing signs. But sequence-wise, they're distinct. Really, lines can have two different types of non-autonomous elements. You can have the broken, fragmented line elements themselves, or you can have the completely distinct sign elements. And sign elements have been very active in the human lineage, and there's lots and lots of copies of them in the human genome. I made a mistake once in an experiment and made a probe for a southern blot that had a sign on it. And I knew something was wrong because the radiation level coming off the final southern blot was way, way too high. And then when I imaged it, it was gorgeous, but I clearly picked up something that was there in a lot more copy numbers than the actual gene that I was going for.
Yeah, isn't that ironic that you would have benefited from masking away repetitive DNA?
Yeah, I mean, this is absolutely. So, if you were to... you think about amplifying a gene or something like that, but often if you're amplifying the DNA, you're amplifying the introns too, or the UTRs or something. And oftentimes there's a little element in there that will mess with your experiment, and because of the copy number, that signal will be amplified way greater than the signal from the gene that you're intending to use in that particular experiment. And the same thing happens bioinformatically, where you could say, hey, wow, this gene is expressed so high, it has 100 or thousands of reads coming from it in RNA-seq. And then you look closer and they're all mapping to the UTR, where there's a repetitive element that's super hot in that particular sample. And it's not the gene itself, but you've gotten, your wires crossed as far as informatically. They can really mess with experiments.
One of the probably the most popular repeat in the sign family is LU. What differentiates it from other repeats? Is that the case that all LUs have the same origin, and what are they famous for?
Alu is the retrotransposon that morphed with a tRNA molecule, is the best way I can put it, which is a pol-3 transcribed product. Now you have a particular small non-autonomous element of an active family, the lines, that was incredibly productive and efficient in duplicating itself to all regions of the genome. So the reason signs have, outside the transposable element community, the reason signs have been picked up by everyone is that if you're studying a human gene, and it really doesn't matter which human gene, because of the numbers, you will probably have signs around or in your gene. And so when people go to study at great detail their particular gene, they end up talking about, well, what's this sign doing? Is this sign responsible for the regulation? Because essentially they're all over the place.
Okay, and the other thing you mentioned was LTR, right? Or long terminal repeats. What are they? This is a family separate from sines and lines, right?
Absolutely. It's the other type of retrotransposon, and these are the ones that are really dominate plant genomes. So human genomes would be dominated by lines and sines, and plant genomes are dominated by LTR retrotransposons and their fragmented derivatives. So these are retrotransposons that encode GAG, which creates a structural capsid, and POL, which is a reverse transcriptase to copy the RNA back into the DNA. And there can be other coding regions in there as well. But that's essentially the key components. And it uses, again, tRNAs and a very complex mechanism to copy itself. It never copies itself, and it's not the simple way you're probably thinking, where it makes one long RNA copy and then reverse transcribes that. Instead, it's a very convoluted process by which it makes a transcript that self-primes itself. But nevertheless, it ends up integrating into DNA, and essentially at that moment of integration, reverse transcribing itself back into the DNA as a DNA copy. And what's interesting about these is most of them have really selectively, for insertion, target other transposable elements or other repetitive regions of the genome. So they tend to create what we call a nested configuration, where it's transposable element inserted into other transposable elements and so on. So it creates this stacking doll metaphor of transposable element nesting. So when you look at complex repetitive regions, you often see this jumbled nested configuration of transposable elements, which is very difficult from an annotation or even assembly point of view to deal with in genomes.
And I think the irony there is that when one repeat inserts itself into the other repeat, that makes it less of a repeat, right? It gives it some unique flair.
It does give it some unique flair, and it might also hamper the possibility of the previous repeat to be properly transposed and things like that.
Yeah, cannibalization.
And another instance of this repeats within repeats are these, so you mentioned inverted repeats, right, at the ends of the retrotransposons. So what purpose do they serve?
Right, so DNA transposons actually have inverted repeats at the end, and it's the same sequence from anywhere from typically 8 to 300 nucleotides in length. That's the same sequence on both sides of the transposable element. It's just inverted, flipped around, and not in what we call a direct orientation where they head the same way. And so these are great because they help us define the borders of a transposable element, if you can find both of those. And they act, in the case of a DNA transposon, as the binding site for the transposase protein, which the autonomous element produces. The autonomous element will grab onto these binding sites and cut there, very similar to how a restriction enzyme cuts, using a staggered cut, and then pick up the transposable element and go insert it somewhere else in the genome, and therefore mobilize that transposable element. LTR retrotransposons, and that LTR stands for long terminal repeat, they have longer repeats at their termini, and those are in a direct configuration, meaning they both point the same direction. And they are necessary for the mechanism of retrotransposition. So you really can't have a functional LTR retrotransposon without those repeats. They're structurally necessary for the process in both cases. So I think of them as often as the Achilles heel of the transposable element. One, it really helps the cell define what is a transposable element to know what to silence. And two, it really helps us identify transposable elements. They can't go too much into stealth mode because of these repeats. There are some transposable elements like helitrons, which are a special type of DNA transposon that don't have these repeat, and because of that, they are way more difficult to identify within the genome and still give people fits with, they think they're studying a gene, but it's just a fragment of a helitron.
So since those terminal repeats act as the target's sort of restriction sites, wouldn't that mean almost by definition that once this thing is reinserted, those terminal repeats would be damaged because the transposase got inside it, and so it won't be able to reproduce anymore?
So the cut is actually right outside the inverted repeats in the case of a DNA transposon, right in the flanking DNA. And what happens is if you make a staggered cut and then you insert the transposable element somewhere else, the inserting DNA is not blunt or not flush at the end. It's staggered. It's an overhang. So when that goes into the new piece of DNA, there's a couple nucleotides there that have to be filled in. And what that does is it creates what's called a target site duplication on either side of the transposable element, which is this often, it's different for different transposable elements, but let's just say 8 nucleotide kind of duplicated restriction site that was once one site and now is 2 sites right on the flanks of a transposable element. So if you want to know, is this a really bona fide recent transposition event, you can look for a target site duplication. And if you can identify it, yes, that just basically happened in evolutionary time. That's really recent.
How can you tell a terminal repeat from this target site duplication? Is one sort of transitioning into the other at the end?
Yeah, I mean, the terminal repeats will be shared among the transposable elements of that family, so those will be highly repetitive. The target site duplication is not repetitive. It is a region of the insertion site DNA that's simply been duplicated on either side. But if you look genome-wide, that piece of DNA will not be repetitive in this family of transposable elements. It's a very fine distinction. It's a very small distinction, but informatically, that's actually very easy.
And so the other question is, if transposase cuts out this element and inserts elsewhere, that's not really copying, right? That's just moving.
Oh, yes. That is called what we call cut and paste. But here's what happens, and this is wild, but people figured this out. 30, 40 years ago. So if you have a DNA that is post-replication phase, so you essentially have sister chromatids in a chromosome that looks like an X, and the transposable element is cut out, the excision site, the site in which it was cut out from, can just ligate blunt right together. Then you have a true excision. But if it's after S phase, you have an exact copy right there to do homology-directed repair from. So it can look to the other sister chromatid, which still has the transposable element at that position, and copy the DNA from there to fill in the gap. So essentially what you're left with is, yes, it cut and pasted, and then at the cut site, it copied from the sister chromatid to refill the space. And I should say that that's figured out by McClintock and her predecessor, or the people that were in her lab afterwards, her trainees, with just an incredible eye for genetics, and not what we consider the molecular biology toolbox that we have now.
Yeah, and McClintock was given a Nobel Prize, wasn't she?
Yeah, McClintock, I should say, McClintock is the hero of the transposable element world. Like, everybody has, their knight in shining armor, and McClintock is it for our world. And her attention to detail is incredible. And her career, probably she could have been awarded 2 Nobel Prizes on totally separate topics. She was awarded the Nobel Prize from work that initiated in the early 1950s, but she was awarded that in 83 or 84, long after the discovery. Yeah, and there's some great stories with, I never personally met her, but I trained with someone as a postdoc that worked closely with her, and there's these stories are stuff of legend in the transposable element community.
I think I've read somewhere that the reason why she was given the Nobel Prize so late was that because she was basically wrong about these repeats for a long time. Like, she called them controlling elements, and she ascribed them the functions that they mostly didn't possess, I think.
Well, in her system, they did have those functions, but those functions were specific to her system. And now, I mean, you're generally right. There's a number of reasons why I think McClintock was overlooked for so long. I think absolutely gender has something to do with that. This was not a time there was many women in science, and she was not a very straightforward writer. What she wrote is very dense and difficult to get at. Then she became frustrated with the system and often was publishing her findings in newsletters and not in the journals of the day, where she was allowed to have the space to go on about what she was researching. And then people simply saw them as junk for a number of years and didn't see them as controlling elements. But we now know there's many examples, particularly in high repeat content genomes like maize, in which he was studying, that transposable elements often are controlling nearby genes, or at least influencing the expression of that nearby gene, where a transposable element, especially a non-autonomous element, you can think of it as just a protein binding beacon. And if that non-autonomous element is near a gene, it's going to bring in new proteins, and those proteins can affect either positively or negatively the level of transcription or regulation of that particular genes. They can act as essentially enhancers or repressors. So McClintock, the idea of controlling elements, not every transposable element is a controlling element. In fact, most of them have no ability to control anything. However, there are absolutely examples where they control quite a lot of development.
Most of the families that we've discussed so far, the SINEs, the lines, the LTRs, these are all retrotransposons, right? So why it seems like retrotransposons are more popular, at least in humans, than DNA transposons. Why is that the case?
One of the cases you can assume is the fact that just now you guys discussed about the DNA transposons, and they have to be at a very specific developmental time so that they can make multiple copies of themselves. Whereas for retrotransposons, no matter when they make a copy, they can insert it anywhere and make multiple copies of them. And also the fact, that's just a side note, the fact that there are almost no active DNA transposable elements in humans. If they were active, then there will be more examples of transposition and sort of this burst, but that we don't see that in humans.
Yeah, there are organisms in which, McClintock originally discovered DNA transposons, and DNA transposons, they're incredibly interesting and their regulation is incredibly complex, but they're not in volume the same level as the retrotransposons, whether in plant genomes with the LTR retrotransposons or in animal genomes with the lines and SINEs. So if you really want to discuss the problems that repetitive DNA create, that's greatest with the retrotransposons and not with the DNA transposons. I should say that for the most part, though, DNA transposons are usually more mutagenic in the sense that when they're active, which is a big asterisk, that might not happen often. But when they are, there's lots of DNA transposons that seem to specifically target genes for insertion, particularly the 5' or promoter regions of genes. And that's a particular place where if you have an insertion, it's going to cause a frame shift or cause a early terminated transcript and create a mutation. So those retrotransposons, which I mentioned, often like to transpose into other repetitive regions of the genome, transposition itself might not cause a big mutation there, but if you start transposing into genes, you do. And that's where the famous mutator family of transposable elements comes from, or mu. Mutator is just because of the incredibly high mutation rate a gentleman named Don Robertson originally identified in a line of corn plants that was coming from this DNA transposon family that was very specifically transposing into and mutating genes.
So I just wonder if the DNA transposons can be more harmful or more mutagenic, and so there's a higher chance of them being selected out of the population, and that's why you have more retrotransposons remaining in the population.
Yeah, there's the potential that they would be under a higher degree of pressure for silencing than the LTR retrotransposons, for example. Yeah.
And then there are the repeats that are not transposable elements, right? So one example is, I think, tandem repeats. So what are tandem repeats, and how do they reproduce, or do they?
Tandem repeats is a broad category, but tandem repeats is the idea is that you have a piece of DNA, not a transposable element, but it could be a genic fragment, it can be an intergenic fragment, it really doesn't matter, but that it becomes duplicated right next to itself. So these are not dispersed repeats like transposable elements often are, but these are in cis or right next to each other, local, and it gets duplicated and they point the same way. It's not an inverted repeat, it's a direct repeat. And these tandem repeats, if it duplicates and now there's two of them in a row, this can often lead to an array of hundreds in a row of the same sequence. Maybe it's 200 nucleotides long, 200 base pairs long, and it's over and over and over again right next to each other, all in the same direction. And these tandem repeats are thought to occur in the process of DNA replication, essentially by slipping of the polymerase, and the polymerase goes back and reads the same region a whole bunch of times and produces this array of repeats. And the interesting thing about them is they happen and they disappear very quickly on evolutionary terms. So evolutionary time. So you might have two individuals of the same species, but that are from different areas of the world, and they have they have the tandem repeats, but in one case, there's 3 copies of the 3 in a row, and in another case there's 100. And so they evolve much faster than genes, so people have used them as markers because it's very easy to determine how large the array is, and it's rapidly evolving, so they can use them as polymorphisms in experiments that have been very useful for mapping disease or traits.
Right, and then there are I think they're called simple repeats or just low complexity regions, which is like maybe the same letter or the same couple of letters repeated many times over. Is that the same mechanism?
Yeah, these are regions. There are low complexity regions of the genome that can happen from a number of different mechanisms. There's mechanisms we understand in which a poly-A tail, which just reads A, can be reinserted back into the genome. And that leads to a region that's difficult to deal with by assembly or by annotation, and that can happen multiple times in the same genome. So now that's repetitive within that genome. There are cases, whether it's ATs or AGs, where there's long arrays of just the same 2 letters over and over again, which is probably, I mean, one way to think about it is it's the same thing as a direct repeat or a tandem repeat that we just discussed, but it's on a much, much smaller level, where the size of the tandem repeat is 2 nucleotides in that case. And it's thought to happen by very similar mechanisms. These are very difficult regions to deal with in the genome on an informatic point of view, because a lot of times they simply will be either masked, or the reads won't be efficiently aligned to those regions of the genome. Or the reads before alignment, the reads are tossed out because the reads itself are a simple sequence, so it's considered an error of sequencing.
Well, let's now talk about your paper, Keith. That's called The Case for Not Masking Away Repetitive DNA.
Yeah, you can call it a paper. I call it a rant, right? I got annoyed with a particular set of papers that came out that was completely obviously ignoring the repetitive fraction, and I could account for what these papers were seeing if I just very simply didn't mask away the genome. And so, that anger translated into my fingers moving, and in about a day or 2, I had written this off, and just on a whim, just boom, here, take it into the journal Mobile DNA.
Yeah, so now it's a peer-reviewed rant.
That's right. And now it's it's like that moment that you might not be proud of that's captured forever in the literature. It's like, oh yeah, I guess I was on a kind of a tear that day. But I'm usually not that opinionated about, how people want to approach their different experiments. But this was a case where it's like, well, gosh, there should be some information that everyone knows about repetitive DNA.
Yeah, and it has a very specific purpose, which I think you state in that note, which you say, like, I hope when people Google how to mask repeats, they will discover this article. And that's exactly what happened to me. So it definitely works in that way.
Awesome.
Can you tell a bit more about that case, like when people could not explain something because they were masking repetitive DNA?
Right. This often happens in our field, or in biology in general, that people are running a standard pipeline of analysis, and somewhere in that analysis, there's what's called like a rep base or repet, or some sort of repeat based filtering that goes on. And what they're doing is they're trying to obviously simplify their results and focus on the genes of the organism. But to focus on the genes, they've excluded the majority of the genome, and often they're looking at those genes, but they're failing to understand why the genes are regulated the way they are, because they're not considering things like introns or UTRs or promoters because they're chock full of these repetitive fragments. People can identify these trends in their data and even publish on them, that as soon as you redo the analysis but undo their filter, take away that filter of the repetitive region of the genome, you see it in a very different light. And in this case, you can see that the conclusion of the paper was A leads to B, and in reality, you see it as something completely different, that A does not lead to B, but this is all a consequence of a very specific view of the genome in your data that was taken.
Yeah, so I think as humans, we tend to view everything in this dichotomous, discrete categories. Like this is a sign, and sign meaning the SINE, the short interspersed nuclear element. And so this is a sign, and this is an enhancer, right? And so we don't usually imagine that they could be the same thing, but of course, signs as part of the genome, they could be co-opted into this evolution and could become enhancers and whatnot.
I absolutely agree. There's this need in our brains to categorize and catalog things, and I think the transposable element community, generally, we've collected a lot of people that love the things that destroy that, right? Like I like studying dogma crushing theories and dogma crushing ideas of, look, there's an element that's kind of, it's not a sign, or it's kind of a combination of these two things, and therefore our classification system to some extent falls apart. And we've collected people like that in the transposable element community, and I think it might not be helpful, but we've created a very difficult situation for people outside the community to try to understand what exactly is going on.
Why is it difficult?
Well, I often get contacted, what's the program I should use? And it's like, oh man, there's pages and pages of these programs. So you have to get very specific of exactly what do you want. And often these people haven't really thought on a specific level what exactly they want the analysis to do. So then we'll say, well, okay, well, here's the options, and there's 8 different options, and they all have different dependencies or different variables that can be considered, and I'll make it complicated, and that's probably deterring them from doing the analysis. Where if there was one grand program to rule them all, to let's say annotate transposable elements in a genome, then a lot more people would do it. But when you look at the available programs, it's a spider's web of all these different people reinventing tools and building tools on other people's, and it takes expertise to do. It's not just a click and run this one little tool and you're done.
So you argue against these people who mask away the repeats, but clearly your position is not that they should just do nothing about the repeats, right? Because you are talking about like various programs to annotate the repeats. So what is your approach that you advocate that is somewhere between ignoring the repeats and masking them away?
Well, if you happen to be in an organism that's well studied, a model organism, a crop species, human, the number one thing I can say is go find the official annotated transposable element, annotation of that genome. It's much easier than producing that yourself. And if every single person or research group annotates their own elements, the problem is they might not make those publicly available. And essentially everybody's analyzing a different set of elements, and that gets really difficult to deal with. So obviously there's not an official genome annotation for every genome that's currently being done. There's lots of genomes that come out almost on a daily basis now. But if you're in human, mouse, Drosophila, C. elegans, zebrafish, Arabidopsis, and many in the plant world, many of these have had major genome projects to annotate the transposable elements. And I'm not saying by any means that those are perfect, but at least we can work on a common set. And so it's not different between each and individual analysis.
Right. So let's say you are working on a human or a mouse, and you have these annotation tracks, but the natural inclination then, since I have these tracks, I'll just use them to mask the repeats away, right? But you are arguing for something else. So what, in your view, should I be doing to my repeats, if not mask them, but still take these tracks into account somehow?
Yes, if you have sequencing reads available, try and map them to those repeats. Many of the repeats have the polymorphism, so that given a long enough read, you should be able to assign a specific read to a specific locus. It doesn't always have to be repetitive. There are different subfamilies. There has been different diversity between the transposable elements or the different repeat families, so that you can say which read comes from where. And even if they don't, then there are consensus elements available, or you can map them to a family and not individual elements, and then can assay if a specific family is affecting the genes nearby it, or overall regulation sort of thing. So you don't have to turn a blind eye to these repeats, but you can do some sort of analysis to see if they have an impact on whatever it is that you're studying.
So if we're talking simply about read mapping, right, you could just map the reads onto the genome, and the genome itself will contain, among others, these repetitive sequences. And if, as you say, they're distinct enough to allow read mapping, then the reads will be mapped, right? So I don't see how having separate tracks for repeats would help here.
It's not a separate track. It's just the annotations are done differently. There are a lot of different classifications of the transposable elements based on families and mechanisms. And so these are just annotations. They just say where in the genome they are.
Right. So I'm just asking, like, what would you use that for? If your subject is not directly related to repetitive DNA, right? If you're looking for just general regulatory sequences, you're saying I should use these repeat annotations. In what way should I use them? Like, instead of just discovering regulatory sequences de novo, regardless of whether they are inside repeats or not inside repeats, just being completely agnostic, you're saying I should look also whether those are repeats, just to know that they are repeats, or what was the end goal here?
Well, let's use a very specific example that might help illustrate what we're talking about. So if you had a gene that you were interested in, and you examined the, let's say, transcriptome in two different, in wild type and mutant, just to make it simple, there's multiple times in that pipeline that people will filter away either the reads itself or mask the DNA that in which the read mapped to of the repetitive DNA. So you could look at a gene and say, look, the counts are identical across the gene, so there's no difference here. Or there could be a case in which you have a transposable element in the promoter, and the transposable element has a number of reads, whether they be uniquely mapping or whether they be multiple mapping, to that transposable element in the promoter, and that might correlate with some effect that you're seeing. So if you're not even willing to see that because either you've filtered away the reads or you've masked the regions of the genome so the reads won't display if they map there, or won't even map there, then you're not looking at the full promoter. You're looking at, let's say, 90% of the promoter.
Right, but if I just did nothing, right, if I didn't mask away the repeats, but I also didn't use any specific repeat annotations, I just did nothing, I just used this stock genome assembly that contains all the repetitive regions right there. Would that be correct, you think? Or should I do something extra to accommodate the repeats?
If you didn't annotate the genome and just mapped everything, it depends how you handle your multi-mapping reads. Essentially, what you're going to be looking at as interesting is all the repeats. I mean, for example, if you're mapping small RNAs and you're not going to annotate the genome, then basically you're just looking at where the transposable elements are in the genome, because those will all very specifically map there, and you're not going to see sometimes these other interesting things that are happening with genes or something. So the annotation can tell you, oh, I'm expecting this peak. It's predicted from the literature, essentially, that these are small RNA generating areas, and I don't need to focus my analysis on what the heck is this thing, right? So the understanding how the repeats, relate to a specific gene, I think is where the annotation comes in. You need to know essentially a couple things. You need to know where the repeats are on that genome track, for example, and you need to understand how the reads were mapped. Because if you're going to say, I only want 100% uniquely mapping reads, in the genome, then essentially you've said, I don't want repetitive DNA to be mapped.
That's a good point. So you're essentially masking away the repeats without explicitly acknowledging it just by setting this threshold.
Correct, except in the examples where maybe there's a SNP or a polymorphism that lets you uniquely map something. So now you're massively underestimating and you're throwing away probably a huge amount of the sequencing reads. The second, if you decide, for example, to map all, so any if something multi-maps, map it each and every time to the genome, well, that's going to massively amplify the number of if you have reads in whatever experiment this is to an ALU element, that's going to massively duplicate that number, that read over a number of different times. So if you're looking only at a specific chromosomal locus, one gene, then you might want to do that because those reads do exist. But if you're looking at a genome-wide level, that's all you're gonna see basically in the genome is the ALU elements and lines. So there's understanding how the reads were handled and how the repetitive DNA was handled, I would say is just as important to be upfront about that and understand the assumptions that were made and the outcomes that those assumptions will lead to than actually considering the repetitive DNA.
Yeah, that's a good point. I think the RNA-seq community is much better about this because they have, for example, RSAM or Callisto. They do this expectation maximization to reassign reads to different transcripts according to the evidence. So they won't get this case where either everything is ignored or everything is amplified. But for example, with the CheapSeq, I don't think they have anything like that. So you're essentially using like a stock read mapping software, and the options aren't as great there, I think.
Yeah, that's right. For something like RNA-seq, Molly Hamill at Cold Spring Harbor created, I believe it's called TE Toolkit, and it's a program that is essentially doing something very similar to DE-seq2 for differential expression analysis, and you can use it for mRNA-seq, or you could use it for RNA-seq, or you could use it for something like ChIP-seq for peak calling, and it's specifically looking statistically at the regulation of the transposable elements in that genome. So there's a couple different assumptions that probably should be made between the transposable elements compared to just doing a standard DE-seq analysis that you would do for the genes because of the repetitive nature. So that's a very useful toolkit that I highly recommend that my lab has used to look at RNA-seq.
Yeah, what I don't quite like about this approach is that you either ignore repeats altogether, or if not, you have to almost become, like yourselves. You have to become a researcher in this field of repeated DNA and transposable elements.
That's what I was talking about. That's our problem in the community that we've done this, right? We have a community of very smart people that has worked really hard, that there's not a lot of consensus in. And so we've created lots of different tools and lots of different approaches. And typically the gene-centric researchers that come to us, they want, what's the one thing? I don't want to ignore these, but I also don't want to spend a month on these.
Exactly, yeah.
Right. So that's I've written up in the past a little paragraph or something, or these are our go-to tools for your initial analysis. But a lot of that also kind of depends on what genome you're in and how repetitive and what type those repeats are. But I think without excluding some of these great programs that people have built, I think there are some really useful tools out there. Repeat Masker, Repit, these are things that have been really adopted by a number of different non-transposable element type labs as either individual tools or pipelines that I think is way better than doing nothing.
Yeah, speaking of Repeat Masker, there's this interesting thing. If you download the stock human genome, then if you actually look at the FASTA file, the sequence, then the repetitive regions are already soft masked. So the non-repetitive are in uppercase, ACTG, and the repetitive ones are in lowercase. But to be honest, I never quite figured what to do with it. Like, are there any programs that take the case into account? Do you know?
I don't think so. I've never heard of a program that took the case into consideration. The stock genome that I work with all the time also does the exact same thing. And in fact, any intergenic DNA they have as lowercase. And then when you get to the genes, it was like, hey, this is important. We'll capitalize this. But yeah, I think there's better things we can go find in the genome besides caps lock.
And do these arguments that you shouldn't be masking the repeats, do they also apply to these low complexity regions? Like if there's a couple letters repeated hundreds of times over, do you think there's still some signal in those regions as well?
Yeah, I this is my personal view, but I tend to think at some point the whole genome gets transcribed, and there's going to be reads in an RNA-seq experiment, or there's going to, maybe not polyadenylated, but those reads, a lot of those reads actually never even make it to the filtering step because they've been excluded earlier on in the pipeline. So those are particularly difficult regions to cover because it's a common sequence error to spit out something like that. So they get hung up in a not passing quality filter type pre-filter before the genome is ever considered.
I also asked for questions on Reddit, on the R Bioinformatics subreddit, and we've got quite a few questions, so I'd like to go through them, and some of them are quite interesting. So the user Versipelis says, I spent some time during one of my rotations analyzing a highly repetitive gene, involucrin. We always went with the philosophy, don't trust any of the submissions for this gene after 2005, since that's about when we expected to see Illumina submissions. And then the questions for you guys. Do you think it's possible to overcome these repetition issues using only short read sequencing? Or do most of these highly repetitive regions require longer reads from PacBio? I don't personally trust Oxford Nanopore, but is it useful in decrypting these repetitive regions when paired with Illumina deep sequencing? What do you think?
I think, first of all, in the question, I didn't really catch in the question when they were talking about submissions after a certain date, if they're talking about like genome assembly or assembly of the gene submissions,
I'm not sure either.
Or submissions of like annotations or submissions of experimental like RNA-seq or something. But from a structural point of view, Illumina sequencing, the de novo genome assembly, so putting together a genome without a previous one to scaffold, is either very difficult or not suggested with Illumina sequencing. They're just simply too short. They're highly accurate, and there's a lot of them, but they're very short. So a lot of times what people have done with Illumina reads is map them back to a scaffold to find SNPs or small polymorphisms. And so if they're building something purely based on Illumina reads, and it's, over a short distance and not repetitive within itself, then there's a chance they could assemble it with Illumina reads. But I think, the hope is that either through Illumina and longer reads and paired end reads and different approaches through Illumina or with the PacBio or Oxford Nanopore, that this is going to be a lot simpler to put it together as a full piece. And even with a higher error rate in sequencing, it typically can fit together much easier in one or two pieces than in, hundreds with Illumina. But as in any sequencing strategy, mixing these approaches gives you the best of kind of two different worlds.
Yeah, and what do you think about Oxford Nanopore? Because on the one hand, these are, of course, longer reads, but they have also this high insertion and deletion errors, and those are especially crucial in the repeats, because then you could misdiagnose the length of the repeat.
Exactly. I think that's why Keith was talking about mixing a couple different approaches. So where they have the advantages of long reads, and then if you have short reads from Illumina, you can go back into those long reads and see where the errors can be, and trust Illumina sequences which have which are less error prone to sort of fill in those gaps or fix those gaps, so that you can trust the overall annotation or the sequence that you come up with.
And then Versipelis asks another question. Is this problem uniquely eukaryotic? I mostly work with bacteria and viruses now, which is the Wild West in many ways. Do prokaryotes or viruses also have known examples of these highly repetitive regions? Has the age of Illumina led us to miss these in lower order organisms?
the answer to the first question, do prokaryotes have repetitive DNA and transposable elements? The answer is a clear yes. There are types of transposable elements in prokaryotics called insertion sequences, or IS elements, that are in bacterial chromosomes, and then as well can transpose into plasmid material and can transpose outside the chromosome as well. So this is not a purely eukaryotic problem. There are certainly fragments of repetitive DNA. I mean, CRISPR itself is a array of repetitive DNA often in prokaryotic genomes.
That's what R stands for, isn't it?
Right. So I don't think this is a purely eukaryotic problem, but the problem is amplified or magnified thousands of fold in eukaryotes, where you start to get these, instead of what are called dispersed repeats, which they happen once in a while in a genome, but they're not clustered. You get these clustered repeats where it's like hundreds and hundreds of repeats clustered all together. Something like in eukaryotes, you get chromosomal knobs and you get pericentromeres, which is the flank of the centromere. And these are physically made out of transposable element sequences. So you can have arrays of huge distances that typically that does not happen in a prokaryotic genome. So it's still there. It's just a lot bigger of a problem in much larger eukaryotic genomes.
Yeah, that makes sense. So if you have a smaller genome, you have a priori much less space for these repeats to occur. And also I think bacteria are more efficient at ditching this junk DNA, although you probably object to me using the term junk DNA.
No, I've learned to live with junk DNA. There's worse things that you could say. But junk DNA, even in the transposable element world, I would see this autonomous transposable element that is, in a genome and has evidence of recent transposition and say, oh, that's a beautiful element, and then see this other fragmented element and be like, oh, that's a piece of junk. So junk DNA, for me, I guess I just see, where someone else sees all junk, I see occasional pieces of treasure and then a lot more junk. But there's so many of these things in such a large volume that it's hard to look through one of these arrays and be like, yeah, this isn't anything except scrambled transposable element sequence. But, prokaryotic genomes, they're under, many of them, I should say, are under constraints for replication time. So they're under constraint to have a small genome. And so that will help get rid of DNA much faster than some eukaryotic genomes, which don't seem to have this constraint. And so the genome is allowed to get obese, and that obesity is often carried through transposable elements.
Right. So maybe they are, meaning bacteria, are in a better position to call it junk DNA because they are actually carrying the cost of it, whereas it's more or less free for us.
Yeah. Well, unless the transposition itself is either causing chromosome breaks or inserting into genes or something like that, where just carrying a piece of DNA that's far away from a gene and not causing any effect is not necessarily, it's just a passenger at this point. It's not necessarily a detriment, and that's why our genome is the size and is the junk graveyard that it is because we're essentially carrying along so much of this fragmented or part of our evolutionary history.
Yeah, I was also thinking it should be possible to estimate the average growth rate of our genome. I saw somewhere an estimate of an extra ALU element per 20 births, something like that. So it should be possible to estimate how many base pairs per generation we acquire.
Yes, absolutely. I mean, there are absolutely mechanisms of DNA loss as well. But it's a dynamic equation. There's also active line elements, the autonomous versions in human populations, which are often called SVAs. So those elements themselves also that you could calculate. I haven't seen the number, but how many elements per human birth or something like that. It's you could do that calculation. I don't know of it offhand that someone's done it.
User Oloaptasis says, I'm currently working on repeats, and what frustrates me is that a lot of papers would reference repeat databases and use them for their analysis. However, after their analysis, they would not submit their repeat libraries to any database. Am I wrong in my thinking that they should upload somewhere their repeat libraries, or is there a reason why people do not do this? And I think this references what you said about like everyone doing their own repeat annotation, right? So here we're talking probably not about like well annotated organisms, but some more exotic organisms.
I think we are of the opinion that if they're referencing an already publicly available repeat database and it's available to everyone, that's fine. But if they actually did the annotation themselves and created these databases, they should absolutely be available for everyone else's use. And if they're not, I think at least if people request to the authors this list of databases, that they should make it available. There's absolutely no reason not to.
Yeah, and I mean, I think we're dealing in a case here which is probably an organism which does not have its own repeat annotation already on like rep-based repository, which I know rep-based, a lot of people have complained that it's typically opened for academics, but is behind a paywall for people in industry. So there's a difference in availability in this case of those data sets. And any kind of informatic file or database that was used as a halfway point. I know there's a big push now to not only publish the raw data and the results of let's say an RNA-seq experiment, but also put on NCBI Geo like the SAM or BAM file that was the in-between file between the raw data and the results, so people can know that if they're using the same raw data but not getting the same result, then this is some sort of variable or something that was taken into consideration during the mapping, or this is the mapping set essentially that was used to create that final conclusion. So I think the same is true for these databases. Typically, it's unfortunate that it's not publicly available, but I would very much hope that once it was requested to the corresponding author that they made it publicly, or that they at least shared it with the requester.
Yeah. So what's the situation with RepBase? So you said it's free for academics, right? Why is there no like completely free database? Just no one bothered to do it?
Well, I think, I think RepBase was early to the game, and these were people in the transposable element field that really knew what they were doing with annotation, and they created a database. And I don't know why it was given different access to industry versus academics, but now that academics have this for them open repository where there's annotation, transposable element annotation information, it would be very hard to duplicate that somewhere else that's completely free and open for everyone, because a lot of people in the field don't see the problem because it's free to them. And it would be very difficult to start, kind of an industry only. So you can't expect these different companies to cooperate in the sense of sharing data between each other to submit their repositories. So RepBase is not perfect, but I see it as difficult to create these other repositories. Just as it's difficult, there's a couple different naming schemes for transposable elements out there, and it's just some people have adopted different ones, and it's hard to tell academics, everybody do this as far as a standard. Occasionally it works, but often, for some reason, people like different programs or people like different repositories. And so the information can be highly spread out. But again, this should all be able to be clarified through an email to the corresponding author and a request. And that person should either point the requester to the publicly available file or provide that file to them. And if that's not happening, then that's not a breakdown of RepBase. That's a breakdown of the agreement that the author signed when he or she published the article in the first place.
And there's a related question from someone named Fatboy93. Can you ask them, meaning you, about their stance on modeling the repeats and annotating them rather than using RepBase, which isn't really open, and what other algorithms methodologies can be implemented in order to mine these repeats? Is it possible to create and maintain alternative repositories that can harbor these information? Something like RED, R-E-D, can be used. So this is, I think, similar to my question of like, why is there no alternative database?
There are a ton of programs out there that will help you identify the repeats in genomes. And I think that a lot of transposable elements or repeat researchers do those, which are not very happy with the quality of the annotation publicly available. So they do it themselves, and for the specific families they're interested in, or genome wide. I think what might be an issue is that it's not as easy as it always sounds to create a very well annotated transposable element database. It's a lot of effort. It takes multiple people to generate that. So just to maintain a free repository available for everyone, that might be a big of an ask, in which different communities have to come together to maintain a repository. And that's, I think, what RepBase has been trying to do that for a number of years right now. Was there a second part of the question?
I think the user here wants to know, are there other ways to annotate transposable elements if you don't take kind of a rep-based approach? And the rep-based approach is going to be to identify repeats or repeat families, very often using closely related organisms, and then identifying the transposable element in this genome, the set of them compared to a known set. There are de novo transposable element identification programs that use more of a K-mer approach. Repit is going to be a suite of programs that could do this without as a kind of completely different approach from something like Repeat Masker. But as far as modeling, I know there are a couple people that have certainly modeled transposable element dynamics, particularly as it pertains to evolution. But I don't know if anyone. I think there are good enough ways to annotate transposable elements using available sequence rather than modeling. But I could be wrong here. It's a big community and people love to create tools in the community.
What prevents someone from downloading the RepBase as an academic for free and then just releasing it? Is it like a copyright concern?
What prevents it is probably something like copyright. Why hasn't someone done it is a kind of a different question. But no, I mean, again, this is not a problem that I typically run into because as an academic, I can,
right.
I know their URL and I can go there and get what I need, but I've never searched around for an alternative source for these files. But typically, with a lot of organisms now, that organism will have an official genome annotation of the transposable element that you can FTP to or something. If you go to the database for that organism, whether that be, Flybase or Wormbase or whatever organism's website, if they have one, a lot of times you can find an annotation file there as well. And that's truly publicly available.
Yeah, I'm just thinking this is more like a copyright geek thinking me rather than a bioinformatician. But of course, depends on the country. In many countries, like for example, in the US, there was this landmark case that facts are not really copyrightable. For something to be copyrightable, it really needs to be like some kind of artistic expression. And so I'm just thinking that these companies, because this is limiting the companies, and the companies with their army of lawyers, they're in a good position to pick up a fight. I don't know who maintains RepBase, but that would be fun to watch.
Yes. And I don't exactly know that the status of RepBase either. RepBase was a great thing that happened in the community from a really prominent researcher who is now passed away.
Oh.
I know the new updates keep coming along. I don't know exactly the individuals that are producing those, but I don't know the future of these. Kaushik and I here are not in the business of annotating genomes. We work on a couple genomes very closely, but there are other people that this is what they do. They wait for the next genome to come out of whatever obscure organism is next, and then work to annotate these genomes. And so there definitely is a community of people out there that are producing the programs to do this. And my hope is that along with everything else, that this is made truly open in the near future.
User Red Concrete asks, "Hi folks, I'm working in humans and I have a historical interest in variable multi-allelic short tandem repeats and would like to get back to working with them. This isn't really a scientific question so much as an opportunistic way for me to find out how you would find full genome that is all known STRs in humans, and also whether you have ever heard of commercial services for genotyping these. I don't mean linkage map, 5 to 10 centimorgan resolution genotyping. I mean comprehensive. I guess the scientific question for you is, if the above don't exist, then why and what are the technical challenges that need to be overcome to create such databases, marker panels?
Yeah, this is definitely a tough part of the genome to work on, because being short tandem repeats and the fact that they're tandem repeats, the idea is it can be very difficult with current sequencing technology to understand if there's one or two of those repeats or 100, but at the same time that's often valuable information because they can be used as markers, and that's why I think the user wants to know about a commercial service for genotyping. So they want to understand the length of these tandem repeats, how many copies there are, because these can be used as a marker system. This is tough. This is something that I think you could potentially do like a local K-mer analysis, where you could see how many times a particular word in this case not only matches itself once, but also matches again in that same sequence. That's one way to look for kind of de novo, look for tandem repeats. But there are a couple of programs out there. TRAP, TRF, and Troll are all examples of different programs publicly available that are published that specifically go out and attempt to identify tandem repeats, and hopefully the programs can then be used once they're identified to then assay them in a database. But I think that database is going to need to be some sort of sequencing-based assay. The way we would do this typically in our organism is to put PCR primers on different sides of the tandem repeat, where they flank on either side and amplify across it, and essentially just look at the size of the repeat. We're not actually sequencing the repeat itself, but just looking at the size of how long it took, the size of the band that from one side to the other. And that we can use as markers, but that requires really specifically looking on a locus specific level at each individual one, and it's hard to do as a panel.
User Cute and Clever asks, I'm interested in the Sleeping Beauty transposons, and now of course I'm interested too. So here she says, if this topic could be elaborated on, that would be great.
Sure. Sleeping Beauty is pretty cool. Again, like I said, you find it, you name it, right? So, Sleeping Beauty is a transposable element that when, there's a specific researcher that this has essentially been his career. His name is Zoltan Ivex. And Zoltan essentially looked at, all of these transposable elements that were either fragmented or mutated derivatives, and lined them all up and deduced from all of these different lined up sequences what must have been the ancestral active element before all these mutations occurred. And then he created that element where there was no active element in this particular genome and put it back in, and that element was active. So Sleeping Beauty comes from the fact that you had all these non-active transposable elements because they were mutated, and then he could create and wake up, I guess, the full active one that must have existed previously to create these mutated copies. So the reason he did all of this is because he realized the utility of transposable elements as tools. They can be used to integrate other things into the genome. They can be used for tools for research, much the same way people are using CRISPR-Cas now as an initial discovery, and now there's all these tools you can generate from it. Transposable elements were very hot for these tools for a long time. And so the idea was having this active transposable element could be used and is in fact being used for things like site-specific integration of material into a genome, or specific mutagenesis. And Sleeping Beauty is a really great example of basic research turned into applied tools.
Did he just publish a paper and say, well, let's call this now Sleeping Beauty? Let's take some guess to say.
Well, right. I can't exactly tell you the history because I've heard, this has happened before I was in the field. But yes, to name something Sleeping Beauty, you would have had to already do it to realize that it can be done. So there must have been a name earlier on, a database type name for these types of transposable elements, and that they renamed Sleeping Beauty.
Okay.
Yeah, this is often transposable elements often have their database name, their official name, and then the cute one that people have called it.
Oh, right. So this is not like an official name.
Well, Sleeping Beauty, I mean, it's probably more official than the database name that no one's ever heard of, right? So Sleeping Beauty is a great story, and it's a great story of what we call making consensus elements. So lining up mutated or lining up all the different elements in the genome into one element, even if it doesn't exist, but that either must have existed or is the sort of average of all of the other elements. And in this particular, we use these informatically, and we use consensus elements quite a bit in our research just to understand, well, what's a prototypical element of this particular family? What would that look like? And in this case, Sleeping Beauty, which is the average of all these fragmented or mutated transposable elements, was actually then put back into the genome and used as a tool.
Okay, so I think the last one, or rather the last two questions, come from the user Cytokindagirl, and she presumably asks, do we have a good idea of the normal amount of variation between humans in repetitive regions? And the second question is also, is there any evidence that cancerous cells preferentially gain or avoid mutations in these regions?
I'm not aware of any specific studies that talk about how much the repetitive regions vary between humans, but I would like to speculate that there's probably a lot of variation in these repetitive regions. Most of the time, people talk about what percentage of genes vary, or what are the polymorphisms available in genes that are different in different humans, but repetitive regions. I mean, you have to imagine the depth of sequencing to annotate properly these repetitive regions, and then to study the variation. It's a big challenge. So I'm not aware of any specific studies. There might be out there.
Yeah, I think they exist. I think the variation that we see in human genomes, you already mentioned, this estimated how many live births, how many, and then you get an ALU insertion. I think the variation in the non-genic portion of the human genome is much, much higher than the genic portion. So I think there's going to be quite a bit of what people in the field would call structural variation, whether it's happening because of a transposable element or not. But this is basically not just, structural variation is in opposition to single nucleotide polymorphisms or SNPs. So structural variation is plus, minus, absence, presence, inverted or translocation piece of DNA. So transposable elements fit into that and are causal for creating those structural variations. And so I think it's a region in which I think it's been studied. I don't think we understand that nearly as much as we understand something like the genic polymorphism level, because when people are typically sequencing human genomes, even in a hospital, in a clinical setting, they're simply aligning those reads back to the genic regions of the genome to try to find mutations that patient might have. But the rest of the stuff is just trash canned, because they're not even close to de novo assembling human genomes on a daily basis. We essentially can sequence and realign or call particular SNPs. So the structural variation of human genomes is probably 5 years to a decade behind the analysis of coding region variation in the human genome.
Yeah, if I remember correctly, microsatellites, which are a type of repeats, they are actually used in criminology and to establish, when they collect the DNA from the crime scene or maybe to establish paternity or stuff like that. So they must mutate super fast, super quickly.
And those are those are things like repeats that can evolve very quickly. And so when you see them on CSI, under this really cool purple light that no lab is actually lit up with, running out their gels and getting results in one day, which again does not happen. And they can essentially get a molecular fingerprint by looking at these highly variable or very quickly evolving regions of the human genome, they're not looking at exons, right? They are looking at intergenic, typically tandem repeat microsatellite regions of the genome, and they vary so greatly that if you look at a panel or a number of these, you can get to the numbers in which this is like a specific fingerprint of an individual. That set of variation at those repeats is not going to be present again because there's not enough people on Earth, right? There's it's a probability game, so therefore they can say that there's a 1 in 10 billion chance that they have the correct individual if you look at enough markers.
Any thoughts on the second question about the evidence that cancer cells preferentially gain or avoid mutations in these regions?
Yeah, well, cancer cells, again, the main databases that we have now, like the Cancer Atlas, they're primarily looking at coding region variation in all of these different cancers. But what we do know is that transposable element activity is closely associated with very specific types of cancer. It's not thought to be the initial primary driver. It's thought to be kind of on a secondary level in which the transposable elements become activated, start mobilizing to duplicate and create the mutations that can, cancer is a road that cells go down that accumulate mutations. And part of that mutation accumulating process can be because of the activation of transposable elements, which will certainly speed up the whole mutagenesis of that cell. So closely associated with transposable elements in cancer, maybe not 100% causal, but again, a lot of times the cancer research that's been done is particularly bad with analyzing just coding regions versus what we hoped in the future, which is the whole genome.
Cool. Any last thoughts? Anything you wanted to discuss?
No, I just wanted to thank you for the opportunity. I think, this is our favorite topic, so we like to sit here and talk about it, but I appreciate that there was interest outside the laboratory and outside the community for consideration and appreciation of transposable elements.
Yeah, absolutely. I think apart from potentially a couple of experts in mobile DNA who may be listening to this podcast, for all the rest of us, this was super interesting, super new, a lot of information to process. So thanks a lot, guys.
No, thank you.
Our pleasure. Thanks so much.